Welcome back, everybody. What I want to do today is connect meiosis, which we learned about in the last unit, with the traits that are actually carried by gametes. So let's get started. And let's do that by reviewing what we know about gametes. Gametes are made by meiosis. Gametes are haploid, meaning that they each get only one of each pair of homologous chromosomes, and thus they can only carry one allele of each gene. And therefore we can conclude that a homozygote makes only one kind of gamete with regard to that trait. For example, the genotype B can make gametes that only carry the big B allele because they don't have the little b allele. Similarly, the genotype b can only make gametes that carry the little b allele. But what about the heterozygote? Well, it seems obvious now that it makes two kinds of gamete, a gamete that carries the dominant trait and a gamete that carries the recessive trait. For example, the genotype B can make a gamete that carries big B, and it can also make a gamete that carries little b. And let's take a look at that in the next slide. So here we have meiosis, and here we have the genotype B with unreplicated chromosomes. Then in the S phase, they replicate. And then they line up at meiosis 1 with the homologous pairs about to separate. And when they separate, you get one of the daughter cells with big B, and one of the daughter cells with little b. Now, I know the clever among you are saying, what about crossing over? Yes, crossing over can create it so this one is big B, and this one is little b, big B. But we're going to ignore that, and I'll tell you why. Because the result is going to turn out to be the same, and you should convince yourself that the result would be the same even if there's crossing over here. So in meiosis 2, the sister chromatids are going to separate. And you're going to get one gamete with a big B and another gamete with a big B on this side. And on the other side, you're going to get two little b gametes. And you should convince yourself that you will have two big B gametes and two little b gametes, even if crossing over occurs. So that's when we're considering a single trait. There are two types of gametes produced by the heterozygote. What about if we consider two traits at a time? And let's say that both of them are heterozygous. In other words, what kinds of genetically distinct gametes can the genotype big A, big B, little b make? Well, it turns out that there are two different cases. First case is when the traits are on different chromosomes, and the second case is when they're on the same chromosome. And we're going to spend most of our time in this unit assuming the traits are on different chromosomes, and we'll see why later on. So, case 1. When two traits occur on different chromosomes, they will be inherited independently of each other. Why is that? Well, it's because of independent assortment that we learned about during meiosis 1. Let's look at an example of that. So, here we have it. Here are 4 replicated chromosomes for the genotype big A, big B, little b. And when they line up at meiosis 1, there is one combination that looks like this and another way of doing it that looks like this. Let's look at the left side only first. In the left side, the big A and the big B have segregated to one side, while little a and little b have segregated to the other side. And they will, of course, after meiosis 2, produce two big A, big B gametes and two little a, little b gametes. But there's another way to arrange to independently assort these chromosomes in meiosis 1, and that's shown here. Big A could be associated with little b, and little a could be associated on the same side with big B. There isn't a third and a fourth way, because if you were to pull this A to this side, this big A to this side, and this little a to this side, and switch the B's, you would have the same thing, just going to different sides. They'd still in the end produce two big A, little b gametes and two little a, big B gametes. So you see that meiosis can produce 4 different kinds of gametes, 2 of each kind, so they each occur 1/4 of the time. So let's now take a look at the second case where the 2 genes are on the same chromosome. If the 2 genes are on the same chromosome, then the alleles that are on that chromosome will tend to be inherited together. So why is that? Let's think about it this way. Suppose flower color and plant height are on the same chromosome, but that for a given plant, one chromosome has the genotype big G, little h, and the other has the genotype little g, big H. During meiosis, big G, little h will be inherited together because they're on the same chromosome, and little g, big H will be inherited together because they're on the same chromosome, unless what happens? Right, some of you have figured this out. Unless crossing over occurs between the locus for flower color and the locus for height. So we're going to take a look at this exact problem in a second. But before we do that, genes that occur on the same chromosome are said to be linked for this reason. So let's take a look. Here we can see we have a chromosome where there's big H and little g on the same chromosome. And here we have one with little h and little g on the same chromosome. When we put them through meiosis, if there's no crossing over, gametes number 1 and 4 will be created most of the time. Because the genes G and H are linked, most of the gametes would be big H, little g, and little h, big G. Crossing over would produce these recombinant gametes, numbers 2 and 3, and they're referred to as recombinants because of the recombination of alleles that occurs during crossing over in meiosis. Because crossing over happens on most chromosomes, two linked genes will behave independently if they are far apart. So if G and H were on opposite ends of this chromosome, then many, many more recombinants would be produced, and you would get a distribution of gametes that is very closely 1 to 1 to 1 to 1, or a quarter of each of all gametes would be each of these genotypes. I'll leave you to think about that and ask questions about that. Because of meiosis, we can calculate the expected frequency of traits and gametes. The term expected frequency refers to the average number or the average percentage of times a chance event is expected to occur. What is the expected frequency, for example, of each type of gamete given the genotype? And you should probably put this in a table in your notes. So here we have 5 cases. If you have one trait that is homozygous, then it can only make gametes that carry big A, and will make it 100% of the time. One trait that's heterozygous, it can make a big A gamete or a little a gamete 50% of the time. Now I'm going to just show you the rest of them. So here they are for the final 3 problems. 2 homozygous traits that are not linked, big A, where they're both homozygous, 1 gamete 100% of the time. 2 non-linked traits, 1 heterozygous, 1 homozygous. There are 2 gametes that can produce, each 50% of the time. And for doubly heterozygous 2 traits, 4 gametes each produce 25% of the time. Pause the video if you're having some trouble figuring that out. Get this down in your notes. I'm going to leave you with these problems for fun. What are the gametes for the following genotypes, and what are their frequencies? You can calculate those on your own time. If you have any problem with them, come to me in class and we'll work them out. So the general rule for calculating the frequency of a trait in a gamete is to, for every homozygous pair in the genotype, replace it with a 1. And for every heterozygous pair in a genotype, replace it with a 0.5, and then carry out the math. So for example, for the genotype big A, big B, little b, the first is heterozygous, so it's 0.5, and the second is homozygous times 1. So the frequency of the gametes is 0.5, and you can figure out by inspection that the gametes are big A, big B, little a, big B. They're equally likely at 0.5, 50% of the time. Here's the doubly heterozygous one. This is heterozygous, so you put a 0.5 in there. This is heterozygous, so you put a 0.5 in there. Carry out the math, 0.5, 0.5, it says 0.25. So these 4 gametes are equally likely at a probability of 25% of the time. And I'll let you figure it out for this one. Stop the video and make sure you understand that one. And then you can do this calculation here for the triply heterozygous one. I want to change topics now and bring up a new vocabulary term, which is the way a trait is manifested is called the phenotype. So in flower color, the genotypes big P, and big P, little p, so the homozygous dominant and the heterozygous genotypes, both have the purple phenotype because the flowers will be purple, while the genotype little p has the phenotype white. So that's an important vocabulary word that you start to work into your usage. Let's remember that in this unit we are studying Mendelian traits, which means that the traits are affected by alleles on a single gene. But most traits are not Mendelian. Traits like height, weight, skin color, hair color, basal metabolic rate, all sorts of traits of most organisms are not Mendelian. They are affected by numerous genes, and when you're affected by numerous genes, the trait is said to be polygenic. The phenotypes of polygenic traits start to follow a normal or Bell distribution, not an either or that we see with simple Mendelian traits, but a normal distribution. Let's take a look at it. In this example, we can see that there are 1, 2, 3, 4, 5, 6, 7 phenotypes going from white to dark red. And this was made by 3 genes, each with 2 alleles. And you can see that with only 3 genes, each with 2 alleles, you get a very close to normal distribution, the normal being this curve, the expected frequencies of the genotypes of the gametes. If more genes and or more alleles affect the trait, then the phenotype distribution becomes increasingly normal. In this unit, we're going to see that there are many variations on Mendelian traits, and they are important to learn. So we're going to learn about what happens when there are more than 2 alleles at a locus. We're going to learn about incomplete dominance, where the 2 traits can either mix with each other or behave as if they're both dominant, and we can sometimes call that codominance. And we're going to learn about traits that occur on the X chromosome but not on the Y chromosome. That's going to be our agenda for the next 2 weeks. That's it for today. I hope that you clearly understand the connection between the genotype of an individual and the frequency with which the different alleles are put into the gametes. In addition, you should now understand that if we're looking at 2 traits simultaneously, how the traits will be carried in the gametes and the frequency of those gametes. Until next time, go outside, look at nature, ask biological questions, come back to class and tell me those questions, and I'll see you soon.